Hello, everyone. Now we move to section 9.2. So this is part two of our series of video lectures in cell signaling. This is actually the middle part. And next part would be section, and final part would be section 9.3. So in section 9.2 here, we are going to look at cell communication strategies between cells depending on how far apart they are. This is particularly true for multicellular organisms where the distances between cells are very different. So we will look at some fundamental strategies that cells use to adapt themselves in terms of their way of communicating in different distances to each other. Your reading assignment here is section 9.2, which is distance between cells, and the reading pages in your textbook are pages 188 through 191. Now, the major learning objective for 9.2 is to be able to compare and contrast cell communication strategies over long and short distances. And the sub objectives under this would be first, relate the role of the endocrine system to long distance signaling. Two, explain why paracrine signaling occurs over short distances. Three, describe how cells can signal themselves through autocrine signaling. And fourth, describe how cell-cell contact can allow cells to communicate. At the end of this section, please try to answer these self-assessment questions. First, what is one way in which endocrine, paracrine, autocrine, and contact-dependent signaling are similar to one another? Second, what is one way in which endocrine, paracrine, autocrine, and contact-dependent signaling are different to one another? As I mentioned earlier, in multicellular organisms, the distance between communicating cells varies. And so physiologists and cell biologists have identified four general types of signaling depending on whether the signaling is long distance or short distance. So first is endocrine, second is paracrine, third is autocrine, and finally there is contact-dependent signaling. So in this section, we will see the similarities and the differences among these four different types of signaling. So the first type of signaling between cells involve what we call as endocrine signaling when the cells are far apart from each other. So this is long distance form of signaling. So in this figure, 9.5A in your textbook, here it is illustrated how endocrine signaling takes place. There is a signaling cell, of course. Recall that we still have the four basic components of cell-cell signaling, which is the signaling cell and the responding cell, and there is the signaling molecule being secreted by the signaling cell and the receptor protein on the surface of the target cell or the responding cell. And so in endocrine signaling, the signal molecule travels through the circulatory system, so it has to travel great distances in the body. So for example, from ovary cells, certain sex hormones must travel through the circulatory system in order to reach its target, which is usually one example would be the target cell would be the uterus in females. In males, testis cells can also secrete a different kind of hormone and again travels to its target cell or tissue. In this particular example would be the bone. And of course, When communication happens, then signal transduction takes place in the target cell, eliciting a specific response. In this particular case, um, some sort of cell differentiation will happen. It's also important to take note at this point that receptor proteins can either be found on the cell surface or it can be found inside the cell or intracellular, as we will see later on. The next type of signaling is called paracrine signaling. This involves two cells that are close to each other so that the signaling molecules need to travel only a very short distance to bind to the neighboring cell's receptor. So this is a signaling that where the signaling molecules can travel distances up to 20 cell diameters or a few hundred micrometers. So it is a very short distance between the communicating cells. Of course, we have the four basic components. Still, we have the signaling cell, we have this signaling molecule, and we have the responding cell or target cell, and we have the receptor molecule here, which is not labeled in this diagram. And there is, of course, this specific response as a result of the signal transduction activation in the responding cell. So here in this figure, we can see that the signaling molecules do not require any sort of a circulatory system to convey the signaling molecule, but simply communicate to each other by the simple process of diffusion. Okay, so in this example, we have the signaling cell uh, as a nerve cell. Nerve cells, of course, can either target another nerve cell, or let's say if muscle contraction is necessary, the nerve cell triggers some sort of communication and response to muscle cells contracting. So there's a nerve electrical secretion, an example. Muscle contraction is an example. And also in embryonic development, many of the signaling molecules involved in paracrine signaling are growth factors that stimulate cell division and development. So this is a very common mode of communication between cells in multicellular organisms. So cell biologists who wished to study paracrine signaling and how this causes cell division managed to grow cells outside the body in culture and allowing them to survive and grow well under certain conditions. And it has long been a mystery where and what sort of signaling um, substance comes from the signaling cell to allow neighboring cells to grow. And in 1974, Nancy Kohler and Alan Lipton identified the first so-called growth factor. And it had long been known that cells could be maintained in culture in the presence of a blood serum due to unidentified factors present in the serum. A serum is simply the liquid portion of clotted blood, whereas unclotted blood has some sort of a medium that we call as a plasma. So the liquid medium in clotted blood is called serum. In the unclotted blood, it's called plasma. And Kohler and Lipton were able to determine that the platelets, uh, as a kind of a cell type in the blood serum, secreted these growth factors. So the first experiment that they did in order to identify this growth factor is to simply compare the response of fibroblast cells. So these are the isolated cells coming from body tissues. Fibroblast is a very special type of cells that usually is responsible for maintaining the extracellular matrix, or what we call as the molecular glue between cells that are dividing in order to form tissues. So they can grow this in culture, and here what they did was to compare the response or the rate of division of fibroblast cells in the presence of either serum or plasma. And what they have seen is that the cells that grow in serum actually grow much faster than those that are grown in plasma. So to Nancy Coller and Alan Lipton, these results suggest that there is something in the serum that accelerates the growth or the cell division rate of fibroblasts when treated with serum. So as good scientists, Coller and Lipton followed up their experiment. And in these follow up experiments, they then sought to determine whether the factor causing the growth was directly released from platelets. So, in order to investigate these questions, they grow cells in the presence of plasma and cells that are present in the presence of a solution of platelet proteins. Now, the cells exposed to platelet proteins, as you see in this figure, grew much faster, at a much higher rate. So they then therefore concluded that the platelet actually secretes a substance that are now suspended in solution, which then stimulated the cells to grow. And through a series of further experiments doing or performing biochemical purification steps, eventually they were able to succeed heroically, in fact, because it was not easy to isolate this biochemically, to purify this growth factor from the platelets. And this is what we know today as PDGF, or platelet derived growth factor. And this purified substance does indeed, in final experiments, show to stimulate cell growth and division. So this is how growth factors involved in paracrine signaling were first identified. The third form of cell-cell signaling is called autocrine signaling, and this is a form of what we call as self-signaling. In other words, the signaling occurs by the responding cells being one and the same as the signaling cell itself, as indicated here in this figure. So the signaling cell secretes signaling molecules, okay, which then bind to receptors on the surface of the same cell and therefore trigger a specific response. And it's important to emphasize at this point that signaling molecules don't always involve causing cell growth. It can also cause inhibition of growth, or even a change in the growth pattern called cell differentiation. So this particular type of signaling or self-signaling or autocrine, that's what this auto means self, can, for example, be involved in the maintenance of a differentiation state during development. So once a state of differentiation has been achieved, like a brain cell or a muscle cell or a nerve cell, then this particular type of cells are maintained in their final differentiation state by the involvement of autocrine signaling. And sometimes, on occasion, unfortunately, cells can also self-regulate themselves to grow continuously and in an unregulated manner. And when this happens, we have this phenomenon called cancer. So autocrine signaling is therefore a very important form of signaling together with paracrine in multicellular organisms during, for example, embryonic development. Finally, some signaling between cells occurs because of direct contact between neighboring cells. So, in this form of communication is called contact dependent signaling or direct signaling. Here, what we have is a transmembrane protein on the surface of one cell in the signaling cell, excuse me, and a transmembrane protein on directly adjacent cells acting as a receptor. So there is no diffusion process taking place in this particular type of signaling where one molecule diffuses from the signaling cell and it gets bound by the receptor cell. So this is a direct contact between two transmembrane proteins found on the surface of two neighboring cells. One example of contact-dependent signaling occurs during the development of the central nervous system in vertebrates. Now, in the developing embryo, some undifferentiated cells begin to express delta cells. These are called embryonic nerve cells. They are not quite differentiated yet here at this point. Um, this signal is received by all the cells directly touching the delta expressing cell. So this is the delta expressing cell through a surface protein called notch, okay, which is this. And as a result, the notch expressing cells become supporting cells. They differentiate during embryonic development to form supporting cells called glial cells and not neurons, whereas the delta expressing cells would develop into a neuron. And so this type of contact dependent signaling allows only a few cells to become neurons, whereas all their neighboring cells surrounding them will become cells that would support them or protect them, like glial cells. So this is a very interesting, unique phenomenon of communication that is direct between two neighboring cells. So we end this lecture video by answering two Reeve questions. So the first one is this. You observe a cell that has a receptor protein that binds a signaling molecule that is produced by the same cell. Now, what kind of signaling is occurring in this cell? A, autocrine signaling. B, contact-dependent signaling. C, paracrine signaling. And D, endocrine signaling. So please try to answer this question using your Reeve account. The second Reeve question is this. Of the answer choices, what is the most likely reason that paracrine signaling does not activate the cell that is producing the signaling molecule? A, the cell's receptors have a mutation. B, the neighboring cells are too far away. C, the concentration of the signaling molecule is not high enough. Or D, the cell does not have the proper receptor. So this concludes our lecture video on section 9.2. We will now proceed to the third and last part of this particular chapter. And this is section 9.3. Thank you.